Okay, guys, we started today's class. Today, we started to study about the antibiotics. And again, so today we are going to start with this the quiz problem. Be ready for this. Everybody, you see that the screen, right?
Yes.
Okay, good. Hey, who is missing?
I'm sorry, the school Wi-Fi is so slow.
Okay, you can see the pin number after this. So anyway, I will start. So pin number is on the bottom here, Sejin, okay?
Okay.
Yep. Okay, we start. Maybe it's a big problem. Guys, we repeated this problem several times. What is the virus size?
100
The size-
nanometer on average.
The average is 100 nanometer. That's some good number as a reference. Good to memorize. Okay, so move on. Sejin, now you are connected?
Yeah, I'm just
Okay.
seeing.
Okay. Okay, this problem also we covered a little last time, and then I gave you some table about this. Membrane, lipid membrane for virus, or maybe lipid membrane for mammalian cell, it may be similar. It can be about 4 to 5 nanometer. We can calculate, but we can do it later. But for now, I think it's a good number to memorize. So 4 to 5 nanometer, the thickness. And move on to the next problem. So here, this is a spike protein.
Professor.
Yep, any problem?
TA Xiao says something on the chat.
Xiao, any problem?
Your microphone is not stable enough.
My microphone?
Yes, it's not stable and has some noise.
Okay. Maybe.
And somebody's microphone also collected some noise about others people's communication.
Okay, maybe other guys, your environment has some background noise. Then when you do not talk, maybe you can turn off your microphone, and you just turn on your microphone when you speak. Xiao, do I sound okay now?
Yeah, now it's fine.
Oh, okay. Okay, here I just showed you the shape, the picture of coronavirus. If you can think, the whole coronavirus size is about 100 nanometer, and those membrane structure is about 5 nanometer, then you can assume what kind of a size of those spike protein. So you can gauge, you can collect all the information you can get, and then you can estimate. This is not the number I gave you to memorize, but from the picture, you can approximately estimate the size of those proteins. So in this case, that protein is a little big protein. As a protein, 10 nanometer is considered to be quite big protein. Sejin, you think differently?
No, I think so.
10 nanometer is a little big protein. Okay. So now we have some picture that 100 nanometer size of virus particle, and then thin layer around there, and then about 10% of the size of a spike protein on the surface. By the way, the coronavirus is corona. What is the meaning of the corona? Do you know?
Means a crown.
It means the crown. You know the crown, right? The head of a king. That crown, corona. So shape of those coronavirus is similar to crown. That's why the virus name was given as a coronavirus. That corona one spike is about 10 nanometer the size. Okay. Move on. Evenly distributed number. Actually, I gave the specific number of the antibody, the length. Anybody remember the exact length of antibody? In the table, in the last week the lecture, I gave you 8.4 or 8.5 length. So antibody is like Y shape like this, and those whole length is about 10 nanometer. So I just wanted to give you the idea. The size of antibody is quite similar to one spike of a coronavirus. Maybe coronavirus spike is a little bigger than antibody, but that's a similar size. Later, we are going to learn about those immunotherapy. One of those main protein structure is antibody, immunoglobulin G. So it's good to know approximate size of antibody and approximate the size of a virus or something like that. Okay. So again, let's think. Antibody size is about 10 nanometer. Virus size is about 100 nanometer. Then you can get some idea, some picture. You know, around one virus, there can be multiple antibodies can bind. If the binding site of coronavirus is the spike protein, how many spike protein were there in coronavirus? Just a guess. How many other spike protein did you see in coronavirus? Just a guessing, guys. Maybe 30, 40, 50 other coronavirus spike proteins you have seen. Then if the antibody bind to the spike protein, you can imagine that maximum binding site of coronavirus is maybe the 50, 30, 40, 50 number. That number of the antibody can bind to the coronavirus. Okay. Actually, this problem, this answer was given in one of your video you have seen. In the video, it's just specifically said about the answer of this question. Okay, many of you gave the answer as a tenfold. Now I gave you the answer as a tenfold or the thousandfold. In the video you have seen, anybody, maybe you remember, in the video, the speaker said, our cell is about 100 times bigger than virus, bigger than bacteria. So it's not clear when you say 100 times bigger, is that length or is that volume? What do you think? 100 times bigger than bacteria? Which aspect is that 100 times? It's a little confusing term, so it can be either the length or volume, but either case, lengthwise we know it's about the tenfold. If it's a volume, it's the 10 to the 3, so that's 1,000 times, right? So 100 is a little weird number. It's just a random number, maybe they just pick up. Mammalian cell is much bigger than bacteria, but lengthwise about 10 times, and volume-wise about 1,000 times. Two of you gave the answer as the 100-fold. Any specific reason? Who gave the answer as 100-fold?
Actually, I miscalculated the tenfold to 100.
Okay.
So,
Who is the other?
Mm,
Someone?
yes.
Any specific reason?
Mm, surface.
Wow, surface area. Okay, if it's a surface area, then maybe it can be 100 times. But usually when we compare those size, it can be either length or maybe volume, I think. Surface area is not common to use, I think. Youngwon, I can see only half of your face, like this. Could you change your angle of your camera? Okay, move on to next problem. Okay, what is mitochondria? Anybody can tell? John, life science major. What is okay, Sejin, sorry, you go ahead.
It is one of organelle for the cellular respiration.
It's one small organelle inside the cell, and mitochondria is responsible for making ATP. You may have heard the name. And mitochondria is a similar size to bacteria. What is the size of bacteria? Doyun, what is the size of bacteria?
One micrometer.
It's about 1 micrometer. So I just changed the word bacteria with the mitochondria. Mitochondria is, we believe that mitochondria was originally separate bacteria, and it enters the animal cell or the plant cells, and then they started to live together, so-called symbiosis. And the size of the mitochondria is quite similar to the bacteria size. Similar things for the chloroplast, 염록체, that is also kind of similar size of 1 micrometer. And that's also, we believe that some bacteria which has a special ability to capture the sunlight to convert that into the energy form. So that is also kind of a similar symbiosis example, we believe. So it's kind of interesting and easy to memorize. Mitochondria, chloroplast, they are similar size to bacteria. Okay, bacteria has 1 micrometer. Size was 10 times longer than virus. So if you fill in the virus inside there, it can be 1,000 times. Okay, clear now? We practiced this over the 2 weeks. Still, correct answer the rate is not so high. I wish you can catch this other number quickly. Then I don't need to repeat this again. Next question. Okay. Yeah, at least the majority of you is got right answer. Human cell is about 100 times longer than virus. So it's 100. 100 to the 3 is 100 by 100 by 100. That's a million. Okay. So now you can draw the picture that in one cell there may be many viruses can invade or can be produced inside the cell. Maximum the million cannot be million, I guess. Maybe smaller, but easily it can be 100,000 or multiple thousand, and then it can explode the cell and then escape. Okay, this is approximate the size we are talking about. Virus versus human cells. Okay, in this week, you guys have studied about the antibiotics and also probiotics. Probiotics is something in favor of us for human, right? Then probio, in this case, probio is in favor for something, then this bio should be human cell, isn't it? Or you can say the favorable bacteria can be probiotic, but if it is an adjective, then probiotic, in this case, the bio should be human. Then next question, naturally, what can be the bio in antibiotic? Okay, in this case, the antibiotic is against, anti means against, right? Pro means it's in favor of or for. So antibiotic means it's against bacteria. Then we can think, who makes the antibiotic? Who produce antibiotic in nature?
Fungi.
Well, fungus can make the antibiotic. Then fungus antibiotic is the weapon to kill bacteria, right? Then there is one bacteria, A bacteria and B bacteria, and B bacteria cannot make the antibiotic against A bacteria. It should be always it should be fungi.
The bacteria can produce antibiotic.
Yeah, bacteria can make antibiotic against the other bacteria. So that's also the antibiotic. Then in the case, there is several different kind of the microprobes, like a bacteria or virus or human cell. Human cell is not a microbe, and fungus. And any materials which can kill some of these, they should be antibiotic, isn't it? In previous problem, we defined the probiotics of the bio is a human cell. Then any material which can kill human cell, it can be also against some bio. In the case, that's us, human cell. It can be also called as a antibiotic, isn't it? Bio can be defined for any life forms. But after this antibiotic is discovered and we use this term is fixed only, usually conventionally, only against bacteria. That's how we use this term. Okay, so when you hear antibiotic, that means antibacteria. It's the same meaning for now. Then, if some material kills virus, then you call antiviral. If it kills fungus, 곰팡이, then you call antifungal drugs. So we specify those the terms. So in principle, original the meaning of antibiotic It means any kind of a toxin which can kill life forms. But we limited their usage only against the bacteria. That's antibiotic. So the topic of this week is antibiotic. That means this is a drug against the bacteria. That's mainly what we are focusing on right now. Okay. Now, current situation, the threat right now, coronavirus, it is not bacteria, it's a virus. And antibiotic is not working efficiently against the virus because the life form and also the function is different. So that's why from next week, we are going to study about antiviral drugs. So before we start the antiviral, we are going to study antibiotic first as a one simpler or maybe the earlier the development. So antibiotic history is much longer. Then we study antibiotic first and then move to the antiviral. Last problem? Uh-oh. Okay, it's 100% the correct answer, but only 3 of you answered. Okay, this problem was given to you, the I-class, the material, and then you tried to make some answers about this, and we are going to review some of your answers. So based on this, okay, move back to okay, in our body. Everybody sees the screen, right? Okay, in our body, we have many cells. Taekjun already gave you the first answer 2 weeks ago. Maybe Taekjun's body is composed of trillions of cells. Trillion is 1조 of the cells. Anyway, it's many, many cells. And also we have many different kinds of cells in our body too. What is the average size of a human cell? Length?
10 micrometers.
10 micrometer.
10 micrometer is the average size of a mammalian cell. Some cells are a little smaller, some cells are a little bigger, but approximately 10 micrometer, let's say. Bacterial size is about 1 micrometer. Of course, some big bacteria, small bacteria, there's some different range. But approximately we can tell bacteria is 10 times smaller. 10 times smaller means we can fill in 1,000 bacteria in one cell. Volume-wise, it's a huge difference. Okay, and virus is again 10 times smaller than bacteria. So by nature, this is a kind of easy the scale, approximate the size of each of them. Okay, this is the last time I may ask you this, but you guys, you may see this the problem again, maybe at the midterm exam or final exam. But try to keep this scale in your mind. That is a very helpful reference numbers. 10 micrometer. And let's say, which cell is the biggest cell in our body? Volume-wise?
Egg cells.
Egg cell, how big is it? Do you remember?
100.
So this is the one egg cell and then the one sperm cell. Egg cell is bigger than 100 micrometer. It's a 150 micrometer diameter. And approximately the sperm cell looks like maybe 60, 70 long. And the head maybe about 10 micrometer or something. Okay, so this is kind of the biggest cell in our body. What is the longest cell in our body?
Nerve cell.
Some of the neuron cells can go quite long in our body. And then these cells all together, by the way, you may have also answered correctly, which is the highest number of cells in our body?
Red blood cells.
Red blood cells.
Red blood cells. How many percent do you remember?
84%.
It's about 80% of the cells. Some people has more blood than the other. So let's just say about 80%. 80% is huge, but the weight of those cells is only 2.5% of the whole body. It's surprisingly small, the smaller than average. If it's 70%, then the weight should be approximately 70%, but it's only 2.5%. That means red blood cell is relatively small, and also it has a quite a unique property. Red blood cell does not have something. In our body, all the cells has
Please.
something. Yeah, go ahead. Who is speaking? Red cell is missing of this some material. What is missing in the red blood cell?
Nucleus.
Clear.
Nucleus is missing. That means the DNA is missing. So the genetic information, whole genome structure is not in red blood cell. The lifetime of red blood cell is quite long, couple of months. They can live, but they live without the nucleus. That means they don't have ability to replicate or the giving the making the stem cell kind of things. And that's why they are very small. It's like almost a specialized machine, biological machine. And the number is the highest in our body, but size and weight is quite small. So in our body, maybe we have trillions of cells. Exactly how many trillions, it's not clear, but 10 trillion, 20 trillion, maybe we have many trillion cells. But in our body, not only we have our own genome-based other cells, we have many guests from outside. So especially bacteria. We have many bacteria in our body. Not only bacteria, we have also different kinds of virus living together in our body. Okay, so how many our own cell or how many of bacteria, nobody knows exactly, but many people say our cell has maybe 10 trillion cells and 10 times of bacteria. That means 100 trillion of bacteria live together. So if our cell and also the bacteria size is similar, then we have 10 times more bacteria than our body. But fortunately, bacteria size is 10% of us, and volume-wise, the 1,000 times smaller. So even though we have 10 times more bacteria, number-wise, then weight-wise, maybe we have 10% of our body is bacteria, we can say. Do we really have 10% of bacteria? We don't know exactly, but the reference people usually cite is this paper made in 1977. And in this paper, the scientist, he estimated the number of our cells as 10 trillion and bacteria as 100 trillion. This paper has been cited over and over. So this is kind of a common knowledge or the common sense. People believe that this is the reference number we use so far. And you guys have studied some of the recent materials and they gave up quite good answers. But Dongwoo gave quite good comparison of previously mentioned some number and then new study number. So I invited Dongwoo to give some brief summary of these other presentations. Dongwoo, would you like to use your file? Would you share your file here? Okay, now you can do it.
Okay, I'll share my screen. So.
Okay.
So I'll give a short presentation as, oh, can you hear me, everybody?
Yeah.
Oh, okay.
Yeah, good.
I'll give a short presentation of how the number of cells in our body have been discussed and calculated by scientists over the past years. So the reference is taken by a single research paper in 2004. And oh, I guess I took the wrong reference. It's another reference by 2016 which is kind of different from what it's said in here. So sorry about that. So anyway, how many cells in our body? So there are known to be two types, which is numerous bacteria and human cells, which is known to be each 100 trillion and 10 trillion. So bacteria lives in almost most parts of our body, including our saliva and colonial tracts and our intestines. And human cells are comprised of mostly of red blood cells and other glial and endothelial and various other types of cells. So scientists have constantly tried to track and measure the number of them. So the original estimate, as shown by the professor in 1972, it uses the total number of bacteria cells, an amount of 10 to the 14th, which means 100 trillion. But the research which I have found out said this is incorrect because it uses that it has a same amount of bacteria in the whole volume of our intestines, and it is that information is actually incorrect because as our intestine go to our anus, it actually has decreasing amount of bacteria. So calculating the results and also revising the amounts of human red blood cells, which is calculated by various researches, it shows that the number of bacterial cells is actually 40 trillion and the number of human cells are 30 trillion, which gives about an approximate ratio of 1.3 to 1. This result is compared by using 20 or 30 years of age of a male, which weighs 70 kilos and 170 centimeters in height. So this is the result, and this is the end of my presentation.
Okay, thank you very much, Tomo. So this is a good summary of those cell number. So for a long time, without real the concrete the proof, we have believed that we have maybe 10 times more numbers of bacteria in our body. The recent study is still it's an estimate, but that's a little more reasonable estimate by calculating those numbers and then compare those with the more conditioned, the better condition, and then calculate it. It's approximately 1-to-1 of those cells. About the 30 trillion, 3 times bigger than before for human cells, 3 times smaller than before for bacteria. So about the 30 trillion is the approximate number of the cells we have. Anyway, still the bacteria number is surprisingly huge, isn't it? And the bacteria is mainly, they are in our guts. Gut means it's from going into here and then going in and stomach and going to the intestine and go out to the other exit. Now they stay in the intestine mainly, and that's highest number of bacteria live there. But everywhere in our body, on the skin and inside the mouth and everywhere we have bacteria. Now let's think. We said inside our body, but the bacteria in the intestine. Inside of intestine is really inside of our body, topologically. Topology means 위상학. Topologically, 위상학적으로 inside of intestine or inside of a stomach, is that really inside of our body? Should we consider that inside of our body or outside of our body? Let's say if the bacteria live on the skin surface, then on the skin surface, should it be outside or inside?
Outside.
Yes, you may consider this outside, right? Just the bacteria is attached here. If a coronavirus is attached here, before it really enters into my body, if it's on the surface, then we may consider that outside of our body. Now, then inside of the intestine, is that really inside of our body? if you think about our body is like a donut, okay? If you take the donut and then inside out, then you can expose that inside of the intestine to the outside. So that means from here to the other end, we have a kind of ring structure, like a donut structure, and stretch out from one side to the other. Actually, that's topologically, that should be considered as the outside. Just the shape of the body, just holding that bacteria in that temporarily the space, and we carry them, but it's like bacteria is on our skin surface, right? But anyway, they are difficult to remove unless we just remove them by pushing out. Then they stay there for a long time. So we consider that they live together with us very closely, but it may not be directly inside our body. Okay, now go back to this slide. Okay, as shown by the Dongho, so we have seen some of these the cell counting, the examples. Now in our body, there are another misconception for a long time. Let's say this kind of a misconception that people say from moon, there is only single human-made architecture we can see. People say one structure, call it that way. Do you know which one is that?
Professor, I can't see the screen.
Okay, sorry. Thank you for notice. Now is that okay?
Yeah, thank you.
Yes.
Can you see the body?
Yes.
Yes.
Okay. People say there is a one the human being made the architecture you can see from the moon. I guess you may have heard that kind of the story. Which one is that? Do you know?
The Great Wall of China.
It's a Great Wall, the Malijiangsong they say, but actually it's not true. With the naked eyes, you cannot see the Great Wall from the moon, but people believe somebody has said that, and then whether that's true or not, people just recite it and then just believe that. Now, similar things we have in our body. Now we have many different kinds of cells, but in the brain we have neurons and also supporting glia cells in the brain. And then people has believed that neuron cell number is much smaller than glia cells. And relative ratio of this neuron to glia, those are supporting cells. Anybody has heard of that ratio? Juan, have you heard? Neuron cell versus glia cells?
No, I didn't. Sorry.
Sejin? No need to say
I
sorry.
didn't.
Okay. So people have believed for a long time, glia cell is 10 times more number than neuron cells. So only 10% of neuron cells in the brain, and then 10 times more numbers of supporting cells are surrounding those neuron cells. That's what we believed for 20, 30, 40 years. Very recently, there's one scientist who came up with a new technology to count those cells, and then figured out, surprisingly, those relative ratio of neuron cell versus glia cells is again 1-to-1 approximately. It's a similar situation of human cell versus bacteria. For a long time, we believed that we have 10 times more bacteria. Same situation here. Neuron cell versus glia cell is about 1-to-1. So if somebody say, and many people believe this is some number, but as a scientist, we should be doubt about that real number always. With the new technique, there's always chance that we can crack those numbers. Always we can get a better estimate. Okay. Okay. Now in our body, as you said, we have many other cells, and during those the problem of the putting up, I asked you to give the numbers and also reference, and you gave some references. And I found several different ways of you are referencing. So one way is you put some site of information, website information here, like this. But website like the Facebook or blog site, it can be quite volatile. Volatile means it can be, can be removed at any time to go away. So after some time point, you may miss those information. So it's not safe referencing. Then maybe another the possibility is referencing this way. Now this referencing is one kind of a digital library site, and then the library has this kind of information. But this is also kind of a secondary referencing. So this is not direct referencing reference the specific paper. This is utilizing those secondary the portal site. So this may not be the professional way either. Another way is maybe using this DOI. Do you know what is a DOI? Have you heard the DOI? For the papers and for some document, this DOI is getting quite more and more popular. DOI is a digital object identifier. So the digital object identifier, it can be any kind of paper or any kind of maybe some music or movie clip. They can get this kind of a specific identifier. Then this is maybe second best way to cite, and this can mean only specific information for the document if it is approved one. So here, this DOI has been used, and then the site for this is bio. Can you read this, anybody? How would you read this? Someone, would you try to read this bio? You don't have an idea how to, go ahead. John.
Receipt?
Receipt sounds like, no.
Archive.
Archive. Bio archive. So archive means you store, okay? It's a collection of those the documents. It's so-called the bio archive. And it says what? Preprint. If you publish a paper, later those the published papers PDF file, usually we call reprint. But this one is a preprint. That means this document is not properly published yet. This is before publication. You can still submit, okay, I have this idea, I have this kind of result. Even though it is a before official publication, I wanted to register this into this site. So this is kind of getting more and more popular kind of publication, but this is not publicly acknowledged publication. Just my claim is there. I found this and I did this work and I wanted to register here. If you wanted to make a comment and you do it, but this is my idea. Of course, there is some dangerous possibility that somebody can steal this idea and utilize this in other the legal or the official publication. Now there can be some dangerous possibility, but this format is kind of getting more and more popular new platform. So in my generation, or at least to me, this I don't feel very comfortable if I have a really novel, new idea, new result. I don't want to expose this into this, but this is kind of you show off some secret into YouTube. Okay, I already claimed here. Many people have seen this. If somebody else claims this in other places, then you guys can prove that I'm the first person who showed this kind of things. Okay, so that means this archive, this collection and preprint, this is also can be a little volatile. If the other people is to point out that this is not correct, and maybe the author can retract, then it can be also easily can disappear or vanish. So then this is also a little dangerous to cite. Then other examples, maybe this is also secondary. oops, sorry. Maybe official way of the citing, the scientific, the publication purpose-wise, usually this format, using author name, and this is a common way of using that journal's information, journal name, and year, and volume, and page number. At least I found one of you, Dongwoo gave quite a good the citation method. This is perfect citation. It has here, this is author name, and year, volume, issue number, and also page number, and DOI together. And also the title of the paper. And this is the perfect citation. This is kind of a formal way. This is the best way to show. And then if you have any kind of, for convenience, if you can put up any kind of a website here, that can be bonus. Okay. When you prepare official document, like your the bachelor thesis or maybe official report, then the order and also those format can be different, but information-wise, year and volume or issue and page number and the original source of the journal, this is the official way to use. Okay, so I leave this as your reference. When you need to write official document, then this is the right approach to use. Okay. Okay, next question I gave you as a mechanism of antibiotics and also resistance. So many of you gave quite good answers for that. We can discuss some of them. But today we have a new person that joined our class. So I asked Young-hoon that to summarize those mechanism of antibiotics and also mechanism of antibiotic resistance. Okay, so let's hear the Young-hoon's presentation. Young-hoon, would you like to share the screen and put up the URL file? Okay, we can see your screen.
Yeah, okay. Hello, my name is Choi Young-hoon, and today I will talk about the mechanism for antibiotics and process of developing resistance. First of all, I will give you the brief explanation of antibiotics. You may all heard about the penicillin once in a while. Penicillin was the first antibiotic that was discovered by the scientist named Fleming. Antibiotics mean the drug used to treat disease caused by bacteria. And for the virus, it is called antiviral drugs that, as the professor talked about this at the first part of the class. There are two types of antibiotics according to their mechanisms. First one is the bacteriostatic antibiotics. Bacteriostatic antibiotics is the antibiotics that stop the functions of bacteria that are needed for their multiplying. For multiplying, bacteria do functions such as metabolism or DNA replication or protein synthesis. Antibiotics get inside of the bacteria, and in each cell, antibiotics attach to parts that do some functions that I mentioned before. For metabolism, for example, there can be enzyme activity, and antibiotics attach to enzyme, so they make them not to act normally. And for the protein synthesis, they can attach to ribosome, or for the DNA replication, also they can attach to the enzyme that produce DNA. So by those functions, antibiotics can stop bacteria multiplying. The second type is the bactericidal antibiotics, and this might be the one that you mostly think about the antibiotics image. in your life. These antibiotics kill bacteria directly. This does this pro-function by breaking down bacteria's cell walls. They usually do this by inhibiting the cell wall synthesis. If cell walls are broken down, then the content inside of each cell swell out of the outside, then and it can't keep its shape, so it dies. To compare only mechanisms, bactericidal antibiotics may seem more powerful and effective than bacteriostatic antibiotics. And to explain about the natural selection process of developing resistance, firstly, I will give you a brief explanation of natural selection. Natural selection means that in one species, there can be many entities, and many entities can be literally different in DNA, and some entities are more adequate for the environment and because of their special part of in DNA. So natural selection means those entities survive in that environment and proliferate and become the main entity in that species. For the antibiotics, there are some bacteria that cannot be that do not get attacked by the antibiotics. Those are called naturally resistant to antibiotics. They survive in this environment and proliferate them. So genetically special or naturally resistant to bacteria. Let me explain some functions how they cannot do not get attacked by the antibiotics. Firstly, they can get antibiotics outside of the cell. When antibiotics get inside of it, they can use their energy and some channels. They can get out of some antibiotics outside of each cell. And second one is the more good one, maybe dinky, because when antibiotics get inside into their cell, they have special enzyme, so they can change the molecular structure of antibiotics. So antibiotics cannot stop the bacteria. So as I mentioned before, they cannot stop protein synthesis or DNA replication or enzyme activity, such things. These are possible because of the special part that they have in their DNA. And let me explain the process of proliferating entity with resistance. First of all, before explaining, there are plasmid, and you can simply think that it's one part of the DNA. Bacteria can exchange plasmid between entities. So the entity with naturally resistant and the entity are not naturally resistant to antibiotics can exchange their plasmid, and the entity that is not naturally resistant to antibiotics can have the ability to be resistant to antibiotics. And second one is transformation, and transformation is the process that the bacteria can get the DNA of dead bacteria and change each original DNA into that and keeps it as each new DNA. This process is called transformation, and so the entity that is not resistant to antibiotics can get the DNA of dead bacteria, which is naturally resistant to antibiotics. So, and the surprising thing is that this can happen between different species. So, if there are some naturally resistant bacteria and if you overdose antibiotics, then bacteria which are resistant to antibiotics can get numerically very fastly. Thanks for listening my presentation.
Okay, thank you, Youngwon. So guys, any question to Youngwon? No question? Okay, then let's review. How many different kinds of the mechanism for antibiotic?
2 types.
2 type means the which one is those 2?
Bacteriostatic and bactericidal.
Well, those are 2 categories. You can maybe classify into 2 classes, but those are not mechanisms.
List
Isn't it?
4.
Okay, 4. Would you tell us? Sejin, would you tell us?
Oh, yeah. First is blocking the metabolism and blocking DNA replication and protein synthesis. And then for the bactericidal, they can directly kill bacteria by destroying peptidoglycan cell walls.
Okay, at least Sejin mentioned 4 of them. I think you have studied those 4. Let's summarize again. One is maybe blocking metabolism and protein synthesis blocking and DNA replication blocking and cell wall synthesis blocking or maybe destruction. So those 4 is common 4 mechanism of antibiotic. Antibiotic means antibacterial, we said, okay? So to kill the bacteria or to attack the bacteria, utilizing these 4 mechanism is a common mechanism so far. And those are classified into the different, the 2 classes. Those 2 are, one was bactericidal, and the other one was the bacteriostatic. And some of those, so now the question is which one is more preferable than the other? And some opinion here. You can see the screen, right?
No, we
Okay.
can't.
No,
You
we
can't.
can't.
Why? You don't see the screen?
No, not.
See.
1 second. What's the mistake? Okay. Sorry about it. Now can you see the screen?
Yes, we can see.
Yes.
So those are 2 categories of those. The 2 categories of those antibiotics. So you simply said bacterial cell wall attacking maybe bactericidal, killing the bacteria, and stopping those growth is bacteriostatic. Says for the other mechanisms. So which one is better? Then maybe you can think of the different ways. So here's just some summary that this is the bactericidal for cell wall and bacteriostatic for DNA replication, blocking, metabolism, protein reproduction. I have a little problem for this word, reproduction. Can you change this word into a different word? Reproduction means, I have my own body and I make my own copy. That's a reproduction, right? So protein is not making itself. Protein should be made from indirectly. So this should be just a protein production like this, okay? So different the person summarized the similar way. So you gave the example as bacterial cell wall as a killing, all other mechanisms as maybe the bacteriostatic the approaches. Maybe it's a convenient classification, but not always. And there's opinion, one of them here, is I think bactericidal is better because it makes harmful bacteria dead, certainly. 확실히 kill. So certainly kill is possible. So that's why I think bactericidal is a better kind of opinion. Then maybe you should attack maybe cell wall. Well, even the DNA replication, let's just think. If you block those DNA replication very early stage, if DNA should replicate, then usually DNA is a double strand and it should open up and then make each strand open, and then synthesis should start there and make a double copy for each strand. If you don't have this, the picture in mind, don't worry, we are going to review this later again. Anyway, if you block this DNA synthesis very early stage, then maybe you can stop their growth. They cannot replicate. But if you attack this process in the middle, DNA is already opened, half of synthesis is done, and that was blocked in the middle, then the bacteria has a 1.5 the DNA. It's a very strange status, very unstable. Then bacteria cannot survive maybe, right? So then, in this case, the DNA replication blocking, it can kill the bacteria. Same thing for the metabolism. If you just stop some metabolism mildly, then they starved, and then they cannot generate energy, and maybe they do not grow, possibly. But if you stop the metabolism too severely, then you can kill the bacteria too. Same thing for the enzyme activity. If it's a critical enzyme, you block it, then they cannot breathe, maybe they die. So that every case is kind of a how severe you are blocking of these mechanisms. Sometimes it can be bactericidal. Other cases maybe it can be bacteriostatic. It's case by case. Now, other opinion, I think bacteriostatic is better because bactericidal can bring about natural selection. Now, this natural selection is related to antibiotic resistance, right? So now, antibiotic resistance, you abstractly, the most of you said, there is some mutation, DNA is changed, and those survived. Those are natural selection is there. Now, this process of antibiotic resistance occurring is a kind of evolution for the bacteria. Bacteria has evolved to survive better. Now, those evolution should have two components. Whether you believe or not, whether you are the religious person or not, and maybe revolution is not something that existed. So everything was created by God. Even though you believe so, there is some, if you define the evolution properly, then we observe the evolution in the current stages and everyday life. Anybody can define the evolution? What is evolution?
Survival of the fittest.
Survival of the fittest is one part of evolution. To get, that's the same term we have used here, natural selection. Natural selection is the selection of the fittest. This is one aspect of evolution. You need something else too. What do you need? The other factor.
Mutation.
Mutations is one kind of diversity generations. So that means to reproduce ourselves, we should copy our genetic information, but that genetic information is not constant. We need to generate some changes on that genetic information. So that is what we call mutation. Mutation has a different kind of mutations. It's a point mutations, or it can be shuffling. Some part of those information is exchanged. So different kind of mutation can happen. Anyway, changes of genetic information should happen. That means we generate our offsprings very similar to us, but slightly changed with many different kind of changes. Two of them should occur at the same time If not, evolution does not work. So many variations is generated, and then the fittest one should survive, and then the other 99% or the 99.9% they die out, and this survived one occupy that empty space. That is evolution. And also that is what is happening in antibiotic resistance. So now in case of bactericidal other cases, if you directly kill the bacteria, and this is a little better summarized the terms. So this is a good answer I just brought up here. Now, if you start to kill the bacteria, then you can remove maybe 99% of bacteria cleanly, but that gives high pressure to the bacteria. To survive, those bacteria generate a lot higher error range of genetic information change. That means a lot of mutations, higher rate of mutations occur. And among them, if you push them very strongly, maybe you can kill them out, then you win. But if very small amount, 0.001% of bacteria survived, then they can occupy that empty space, and then they rebuild their community with resistant bacteria. So people are arguing about this. If you give the bactericidal drugs, if you kill them all, then that's okay. If you fail to kill them all, very small amount of the bacteria survive, then there is a very dangerous possibility that resistant bacteria can come back. That's why it's a very bad habit. If you take 1 week of the antibiotic prescription, if you stop eating those drugs in the middle of those treatment, you just eat 3 days drugs and then you don't eat them out, then there is a very good chance that majority of those bacteria died, but still there is a very small population that survived. Then they got trained to resist to that drug. Then after 2 weeks, they can come back with the resistant bacteria. That's the dangerous part. So if you don't kill them all, then it's better you leave them together with them. Just don't let them grow fast enough. That's the bacteriostatic, antibacteriostatic approaches. So there is a 2 argument to that, whether which method is better or not. But so far, seems like the chance of getting the antibiotic resistance is coming from bactericidal drugs rather than bacteriostatic drugs. Okay. Oops. Move forward. Now there's another term for the targeted versus broad. So which one is more targeted? Well, one person put up this comment. I think bacteriostatic is a better approach because it can be more specific. That means it is more targeted. Is that true? Bacteriostatic, we said, it can be metabolism blocking, protein synthesis blocking, or DNA reproduction blocking, replication blocking. Is that specific for one bacteria than the other? Well, in a sense, if you can find the metabolism very specific for only one other bacteria, maybe okay. But protein synthesis, DNA replication, this is a basic function for every life forms. So that means it's quite difficult to make it specific to one bacteria. Okay, so that means if you use this kind of drugs, it's very possible you suppress those growth of those bacteria, bad bacteria, and also probiotic bacteria. I said probiotic bacteria. Probiotic means that biotic is our body. Probiotic bacteria, those are good bacteria. Both of them potentially can be affected together. So to do this really specific, it's difficult to say whether bacteriostatic is more specific than bactericidal. Both of them, you should find specific target and make it specific. Now, this term, gene versus genome. What's the difference? Can you explain what's the difference between gene and genome? Don't be shy. Takchun, what is the difference between these two?
Oh, I don't know exactly.
Really?
In English terms.
Okay, in Korean terms then?
Gene is simply 유전자.
Okay, genome is?
Genome means 유전체, I think.
What is 유전체?
유전체 means group of gene.
Yes, collection of all genes together. Not only just the group, it's all together. Then combined all together, we call genome. Then this can be human genome or bacteria genome, we can call. So this all may be coming from omnibus. It's a collection of a whole thing. That's the genome. This kind of term is extended to different the field. Microbe is the one small bacteria or small microorganism. If it's combined together, all together, what would you like to call? You ever heard of this term in the video you have seen? Microbe?
Microbiome.
Microbiome you have seen. So that's why we have many other bacteria live together with us. So we call this as a second genome, even though those bacteria gene or bacteria genomes are not from our the father and mother, but they lived together with us for long. Even you have seen those the video that when the baby is born, they get a bacteria shower when they are born through those the path when they are born through the mothers. Then this kind of bacteria exposure and they're utilizing those the genome of those bacteria is quite important. And then sometimes you have seen the video that you need to get healthy bacteria the genome. That's why you take those stool introduced into the body, right? In the video, they showed that new bacterial source can be introduced from the other end, but actually you can take it as a capsule. So you can fill it up, the stool in the capsule, and you can eat it. Then either way, go this way or from the other way, the other end, it doesn't matter. Anyway, it can be introduced into the body, but topologically, as we said, this is still outside. It's kind of the intestine or the area of our body. Okay. One person gave this term of fusion that bothered me. By fusion with some sites that bacteria has, the bactericide, antibiotic, fuse with the bacteria. Fusion is this kind of thing is a fusion. But when we say the antibiotic, so let's say this left side, this is an antibiotic, then most likely it happens like this. Then fusion may not be the right term for describing this. What would you like to call? Fusion may not be the good term to describe this. Then how would you like to call?
Mesh.
Match is still a little abstractive. Other term, physical term.
Attach.
Attach is better. Usually we call
Combine.
this bind. Combined is not bad, but usually we call this is a binding. So in this case, we call maybe this is a ligand and this is a receptor, kind of a term we are going to use later. But anyway, this event is this guy bind to this. So attach, combine also that makes sense, but usually we call using this term as a binding. So binding constant, binding event, that's what you say, rather than fusion, okay? Okay, so today's class is over here. So next class is on Thursday, and then assignment 3 deadline is tomorrow morning, the Wednesday at 6 a.m. Sorry, I didn't leave the time for the questions. If you have an urgent question, you can give 1 or 2. Okay, if not, then still you are open to give me the questions through Kakao email, and we meet on Thursday. Okay?
Okay.
Okay, guys, thanks. Thank you, Youngjun.
Thank you.
And Dong.
Thank you.
Thank you very much.